And nice to meet you, everyone. First, thanks to the committee to give me this opportunity to present my work at conference. And this is also my first time to give presentation about my work. Today, I would like to show you about the mechanism linking microbial biodiversity and pathogen suppression. And I'm from China. As you know, China has great population that you cannot imagine. So who will feed China? We need a lot of food production, and also there are, And also there are lots of plant diseases happened in Chinese field. Especially there are concubar wilt and Rostonia wilt. And every year there are 100 billion loss because of plant diseases. Sorry, please wait for a moment. And plant diseases is a threat to food safety. So not only farmers, but also scientists call for a sustainable way to control plant disease. But how can we do? A lot of researchers show that rhizosphere microbiome has strong linkage with plant health and soil health. And theoretically, rhizosphere microbiome can compete with pathogens in soil. And there are two kinds of mechanisms of competition. One is nutrient competition, and the other is a technism. Nutrient competition is the way that bacteria direct grab resources from rhizosphere that to compete with pathogens outcome. And a technism is indirect way that bacteria not only use resources in rhizosphere, but also produce antagonistic substance that will do harm to pathogens. But no matter what kind of mechanism it is, the most vital thing in rhizosphere is the resources. That is what we call the root exudates. And bacterial community can use root exudates very quickly, efficiently to strengthen themselves and feed themselves. That if their invaders outside want to attack them, they will build their great wall to fight against with pathogens. But how can we recruit our rhizosphere community, or how can we organize community? In ecological way, we call it biodiversity. And biodiversity is a controversial topic in many researches with plant disease. And this is the paper cited from former speaker. With the increase of diversity, the pathogen cannot survive very well. It is the positive effects between biodiversity and pathogen suppression. But things will not happen always in the same way. There are also negative effects between biodiversity increase and pathogen suppression, because with the increase of diversity, the antagonistic interaction between communities will also increase. That will decrease the pathogen suppression. So we may ask whether microbial biodiversity is beneficial to our ecosystem and to our plant health, and whether we can use microbial community to make our plants from getting diseased to use them in Chinese field? If so, what is the mechanism behind it? So we use several species of non-pathogenic ones as model strains. The strains is isolated from greenhouse in China, and none of species has a mechanism between pathogens. And some non-pathogenic species, we call NPRS for short, can efficiently reduce plants from getting wilted. And what we can do with our NPRS species, we use 5 kinds of species to combine them in diverse way, including all 40 possible treatments, and use pathogens Ralstonia as model invaders. We tagged Ralstonia with red fluorescent protein that we can track the pathogens, and also we can measure the density of the pathogen with the fluorescent. And what happened in the greenhouse experiment, we divide the whole stage of plant growth into 2 parts. One is latent stage, that bacteria will colonize, that pathogen will colonize in rhizosphere and grab the resources and make a good preparation for big explosion next stage. And in acute stage, the plant will get wilted very quickly and in very severely way. And compared with the pathogen treatment, the NPRS treatment can strongly reduce the pathogen suppression with the high richness of our species. So we may ask whether we can use the increasing NPRS community to compete with pathogens with resources in rhizosphere, and also whether with the increase of our diversity, the intern competition will also increase, so it might be weaken the competition with pathogens. This is the 2 aspects we want to dig further. And first, we use niche overlap to represent the computation ability of NPRS with pathogens. And we use 48 carbon resources to mimic tomato root exudates and to see whether our NPRS strains and also pathogens can use each carbon resources. And if they can use it, the well will turn red. And niche overlap index is to describe the resources that can be used by NPRS to pathogens. And this picture is the carbon consumption patterns of our pathogens and NPRS strains. The patterns of pathogens represents in red, and NPRS patterns represents in black. And this area represents the high niche overlap between NPRS and our pathogens. The high niche overlap is to describe that our NPRS community can use nearly all the niches and all the resources in our ecosystem, that no more excess niches and resources would be left for pathogens, that pathogens will have higher competition with our NPRS species. And this area represents the low niche overlap between NPRS and pathogens. And low niche overlap means NPRS and our pathogens can live in peace without competing with resources, and more weakened niches would be left for invaders to attack the ecosystem. So lower competition is between the NPRS and pathogens. In this way, NPRS cannot suppress our invaders successfully. And next, we use our NPRS to combine into different communities, and we select our carbon resources into 20 treatments to see what happens to our NPRS community and pathogen invasion. And we culture them in microtiter plates. The results show that with the high diversity, there are also high niche overlap between NPRS community and pathogens. And also with high niche overlap, the pathogen can be under control. And next, we use trophic network, the nestiness, to represent the intern computation within our NPRS communities. And nestiness is a kind of way that represents the degree of deviation from fully nestiness model, which means if we calculate a higher value of nestiness, it means that they have higher deviation from fully nestiness model, so which means the lower nestiness in our NPRS community. And this is the similar work which has been done by others. It shows that the diversity have positive effects with nestiness, but nestiness has negative effects with the persistence in ecosystem, and the thickness of the errors is scaled to standardize the coefficient of path analysis. And we also use the same picture to represent and to analyze the nestiness in our NPRS community. In this area, it represents the high nestiness. High nestiness just represents there are high intercomputation within our NPRS community. And it represents the destabilized community of our NPRS. And this area is to describe the low nestiness of our community, representing low intercomputation. And also it describes how our community can well organize themselves to fight against with pathogens. And here we back to the results of our greenhouse experiment. In latent stage, the most important thing is that how bacterial community can organize themselves that built the first great wall to fight against with pathogens want to attack the ecosystem. So in latent stage, the nestiness is the main factor that contributes to the plant health. And the low nestiness means community can organize themselves very well that can make the pathogen suppression. And in acute stage, the high niche overlap contributes more than the nestiness to plant health. So nestiness is a prediction that to plant protection. And here I want to make a small summary about my work. We use trophic network to predict our pathogen exclusion and to show people how we can organize a well community to fight against with pathogens outside, and to make such a guidance to people as applicant to how we can use different kinds of species to combine them to fight against with plant pathogens. And high niche overlap is the main factor that bacterial community can use in this way to have nutrient competition with pathogens. And we combine the niche theory and the networks which can provide the framework to tell people what is the perfect combination of our bacterial community and how bacterial community can fight against with pathogens. And at last, I want to thank my supervisor in Nanjing Agricultural University. And this work has been done all in my university. And my supervisor, Professor Shen, Professor Xu, and Dr. Wei. and also my hosting supervisor in Utrecht University, Dr. Alexandra Rosette. And also thanks to some other colleagues and foundings in China. And thank you very much.
Thank you. Are there any questions for Tianjie? Yes, back. Thank you.
Thank you very much for the talk. I have a question of understanding. What does nestedness mean for the bacterial community? They are using the same kind of resource? Because I do not exactly understand the figure you show.
Yeah, you ask the carbon use patterns, whether it represents in nestedness, right? Yes. The carbon use patterns represent in niche overlap. The niche overlap represents how bacterial community, how many kinds of resources that can be used by bacterial community to pathogens. But nestedness is a kind of model that shows how bacterial community can organize themselves, not the way to show the carbon consumption patterns.